Thanks again, Sarah, for that really amazing introduction, and I want to say thank you especially to the SSE and the ASN for these awards. It's really a huge and surprising honor. And I'm very excited to get to be here today to share my work with all of you, which is going to be about the biogeography of genetic diversity and its conservation. Let me start the timer here. Okay. So I'll start, we'll just talk very broadly about biodiversity. So this is a complex and hierarchical concept. We can think about and quantify biodiversity in many different scales, at many different levels of biological organization. But very often, the thing that gets focused on the most is biodiversity in terms of species, specifically species richness. This historically is because we have a lot of knowledge and data at this level of biodiversity, but this kind of focus at the species level doesn't lend itself super well to a very comprehensive understanding of biodiversity, because if we want to understand what's going on at the species level and above, we also need to know a little bit about what's happening at the population level in terms of different processes that can be sustaining biodiversity across this entire hierarchy. And so in this talk today, I'm going to focus on genetic diversity measured at the population level. Genetic diversity is very interesting in its own right. Of course, it's the fuel for evolution and adaptation. It's very important for populations to have the kind of potential to be able to cope with environmental change. This lends also to their resilience and capacity for long-term persistence. And so genetic diversity is very important for kind of resilience and persistence at the population level, and of course populations being able to be flexible and last for a long time will also sustain biodiversity at these higher levels. And so typically when we look at and think about biodiversity from a kind of macroecological or biogeographic perspective, because we're asking questions at the species level and using species level data, there often seems to be an approach of top-down inference where we're starting by characterizing patterns of biodiversity and then speculating about the different kinds of processes that could have generated these patterns. And in this talk, I'm going to flip that around and start by thinking about the kinds of population-level processes that might then scale up and lead to macroecological patterns. The overall theme of the talk is going to be asking whether there are general rules governing the organization of biodiversity, which is a super huge question, but something that I'm trying to tackle by taking this approach, thinking about the kinds of contemporary population-level processes, so microevolutionary processes acting at relatively local scales, how these might scale up and aggregate across species and spaces to generate patterns that we see in macroecology at a macro scale. And so if we want to think about, what are the sorts of processes that we think could be very applicable across kind of any kind of species and any kind of system, we're going to start thinking very basic. And so the questions that I'm going to talk about today are really going to be focused on very fundamental characteristics of populations. So this is thinking about their size and connectivity. These are ecological features of populations that are very intimately connected with evolutionary processes. These are namely genetic drift, so the random fluctuation of allele frequencies over time, and gene flow, sharing of alleles across different populations. And together, these ecological and evolutionary processes can shape the distribution of neutral genome-wide genetic diversity in populations. And so we are interested in the factors that shape genetic diversity, but we can also use genetic diversity estimates as a tool to then kind of think back about the different kinds of processes and the balances of different evolutionary forces acting at the population level that could be important across species. And as I'm sure you will come to see over the duration of this talk, I'm going to talk a lot about genetic drift and effective population size, and kind of really focusing on this as a kind of critical factor that could be something that is contributing to the organization of biodiversity from continental to potentially global scales. So the couple of different studies I'll walk you through in this talk are all going to take this similar kind of form, where we're going to think about the ways that environments are shaping the sizes and connectivity of populations, what that in turn means for their genetic composition and the different types of microevolutionary forces at play. And then we're going to take a next step and kind of scale that up to thinking about how this could possibly be related to species richness and other patterns of recognized in macroecology. So you might look at this and say, well, Chloe, this looks like your typical landscape genetic study. It's not maybe the most revolutionary thing, but the kind of next step we're taking here is to really synthesize the processes happening across all these species to make some kind of generalizable inferences for macroecology as a whole. But typically, these types of questions used to be a bit restricted at the scale that we could ask them, because this, of course, requires a lot of data. So as you can imagine, this needs sampling multiple individuals in multiple locations for multiple species across their entire ranges. This kind of effort and time and money really builds up. Luckily, evolutionary biology was amongst the earliest disciplines to really embrace a culture of data sharing, and especially with the increase in journal mandates for open data, genetic data have really been building up in online repositories for decades. And now is really a kind of ripe time to take them and repurpose them for different types of questions. And this is really the bread and butter of a field that's now being called macrogenetics, which is essentially big population genetics. We're asking population genetics questions at broad spatial, temporal, and taxonomic scales. There's a lot of different ways you can do macrogenetics. Some flavors of it do direct data synthesis, where we're working with genetic data directly. Alternatively, you can take a more meta-analytical approach and take estimates of genetic diversity and other genetic summary statistics from summaries in the literature. And I've been building a macrogenetic database since the start of my PhD. It is tentatively called MPOP, and I started collecting data for terrestrial vertebrates from the US and Canada, and I have since, with the help of many collaborators and students, both at the University of Manitoba and in my current institute at IDIV, we've been kind of trying to pull all of the microsatellite data that we possibly can, first off of Dryad, and then once we're done with that, we'll try to move on to other public repositories. And the database at the moment has a little over 3,000 different locations, 262 species, and over 150,000 individual genotypes. And so, as I said, I've started with terrestrial vertebrates, and I've been very slowly poking away at kind of expanding this database past the US and Canada. And other colleagues are also starting to collect data from different parts of the world and in different types of taxonomic groups. So just looking at this map here, each point represents a location where we have multiple individuals sampled. And an important thing to note here is that for each of these different studies, we're working with the genotype data directly. So we're able to estimate kind of whatever summary statistic we want, regardless of whether it was presented in its original publication, and we're able to do that in a way that's consistent across all of these disparate data sets. And so to give you a quick overview of the structure of my talk today, we're kind of going to follow a rough spatial gradient as well as a gradient of human impacts, I guess is the word. So we'll start by thinking about general macroecological patterns and thinking about the kinds of population processes that might have led to these, and then we'll kind of work our way down to thinking about urban evolution and kind of what this means for biodiversity change given human-caused environmental change. And in the last part of the talk, we'll go a little into using macrogenetics for applied conservation. Oops. Okay, so the first part kind of concerns the latitudinal species richness gradient, or latitudinal biodiversity gradients in general, which is probably the best known pattern in macroecology. So this is just where we tend to have higher numbers of species towards the tropics. And despite being very well known and very well studied, there's still over, I think, 30 hypotheses proposed to try to understand where this pattern came from and how it's being maintained. But there's no real consensus about, there's no consensus across the different hypotheses what could be the major factors leading to this. However, we do know that many facets of biodiversity do tend to co-vary spatially with species richness, including functional diversity and phylogenetic diversity. And it's been kind of thought that genetic diversity would follow a similar pattern, but we really weren't able to test this at the same scales in macroecology because of these data limitations that I've just talked to you about. And so once we had our data set together for mammals in the US and Canada, that's what we have on this map here, we wanted to explore these continental patterns in genetic diversity across all of the species in our data set, which was around 40. And so all of the dots on this map here are locations where we have genetic data. And to be able to compare it to species richness, we first roughly estimated species richness at each of these spatial locations using just the number of ranges overlapping each of these points. And then we've used an analysis called distance-based Rand's eigenvector maps, which pulls out just the purely spatial variation in the data. And we've mapped the very broad-scale spatial patterns here. And we can see that this technique has captured known patterns of species richness in this part of the world. So for example, we've got very high mammal species richness in the southwestern part of the continent. And when we do the same thing for genetic diversity, this is what we saw, which at first to us was quite surprising because I think the thing that stands out first is obviously they look to be the opposite. So they're negatively correlated, which is not what we were expecting. So I'll point out just, again, the southwestern part of the continent where we see relatively lower genetic diversity where in the species richness hotspot. And when doing the same thing for amphibians, the pattern is a little bit less clear, but we can see it popping out again along the east coast. So the southeastern United States is a global hotspot of amphibian and particularly salamander species richness, and in this region, we also tend to see relatively lower genetic diversity. And so, of course, seeing this result was, as I said, a bit surprising and a little bit puzzling because we did have a priori expectation that kind of all levels of biodiversity are going to have the same general kinds of patterns. And so to dig into this a little bit further, we borrowed a few hypotheses from the macroecological literature. These hypotheses are both based on ecological limits, so they're based on size and connectivity of populations. Is there a concert next door? So the first one of these is the more individuals hypothesis, and this kind of simply suggests that in areas with higher resource availability, we're able to support larger numbers of individuals, and this in turn means we'll have bigger populations and bigger communities. So from this hypothesis, we would expect to see higher genetic diversity and higher species richness, and that the two will be positively correlated. A second hypothesis we explored was related to environmental heterogeneity, and this one posits that essentially more heterogeneous environments have a greater number of ecological niches that species can exploit. However, because species are now kind of specialized to the specific resources of this niche, we're kind of packing more species in, each individually with smaller population sizes. And so from this hypothesis, we would expect to see greater species richness, but lower genetic diversity. And so these two facets of biodiversity would be negatively related. And as we saw from the map, there was a kind of very clear negative correlation, but that was really in the species richness hotspot overall. These patterns are more nuanced, and I should say that these hypotheses are absolutely not mutually exclusive, so both of these processes can be occurring at the same time to shape biodiversity at both scales. And so we tested this using structural equation modeling, which allows us to test a kind of network of hypotheses. So we could test both of these processes occurring at the same time to compare their relative strengths. And I'm not going to go fully into the details of the results here, but just share the most important parts with you. From the more individuals hypothesis, we were looking at the relationship between the availability of resources and genetic diversity and species richness. And here we found that both genetic diversity within species and species richness tended to increase with increasing resource availability. So check. From the heterogeneity hypothesis, we measured environmental heterogeneity using land cover diversity at each of the locations on the map that I showed you. And we found here that genetic diversity was, in fact, negatively related to environmental heterogeneity and positively related to species richness. So we did find support for both of these hypotheses and both of these mechanisms at play in generating these biodiversity patterns across many levels, which together suggests that resource availability and heterogeneity, so basically the availability and the distribution of resources in area, can be kind of basic factors of environments that are shaping multiple levels of biodiversity at the same time. And this is a kind of nice finding that will form the basis of our next pattern that we're going to look into, which is asking, again, the super broad question of why aren't species everywhere? So we were just talking about latitudinal gradients, where we tend to have more species in the tropics than at the poles. So obviously biodiversity is not uniformly distributed across the world. And one way we can kind of categorize the distribution of biodiversity is to think about the regionalization of biodiversity. So biogeographic regions of the world are regions that have more or less distinct species assemblages. Excuse me. And the kind of puzzling question here from a genetic perspective is, why do we still see these regions being maintained, essentially? If a population has enough genetic diversity, in theory, it should be able to continuously adapt and be able to expand its range to a point where we might see these regions start to homogenize. And so the question becomes, kind of what's going on at these transitional zones between biogeographic regions that stopping species from considerably expanding their ranges into neighboring biogeographic regions. But to be able to answer this question, we first have to think about these species ranges. So as I've just said, if enough genetic diversity exists, in theory, we should be able to adapt to anything and see continuous adaptation across environmental gradients. But given that species have finite ranges, this is clearly, not the case. And so asking about the distributions of species and why species have limited distributions was a major question early on in macroecology. And it was Brown in 1980 who kind of formalized this central marginal hypothesis, which suggests that the environments near the center of a species range were the most well-suited for a species. This is where they're able to attain very large and kind of well-connected population sizes, so they have very high abundance towards the center. And then as we move across a range towards the range edges, the environment is changing. It's getting a little worse for species, and so population sizes are declining until a point where we reach, what becomes the range edge is essentially where population sizes or abundance have decreased to a point where, the species ceases to exist, and this is the edge of the range. And so when we're talking about gradients in population size, this is, I think, immediately interesting from a genetic perspective, because population size is quite key for thinking about microevolutionary forces, and specifically the strength of genetic drift. So we know that small populations and big populations can have different evolutionary trajectories due to the differences in the effects of genetic drift. So genetic drift tends to be very strong. So you can imagine in a small population, you move one individual, you add one individual, that can change your allele frequencies quite a bit. And so in a small population, genetic drift is very strong, and this means that random allele frequency change is kind of the dominant manner of allele frequency changes. And that means that we know from theory that then natural selection necessarily becomes less efficient. So when this randomness is the major thing driving evolution, selection, it can still operate, of course, but it might take a longer time for populations to be able to adapt to different kinds of environments. And this is in contrast to what we would expect to see in a larger population where genetic drift is very weak or negligible. And in these instances, natural selection can act very efficiently, and so adaptation can happen on much shorter timescales. And so when we now reconsider this central marginal idea from a genetic perspective, what we would expect to see are towards the range core, where environments are suitable, where populations can get really big. We will see more genetic diversity, populations will be more connected to each other, and then as we move out towards the range edge across an environmental gradient, population sizes are declining and they're becoming increasingly maladapted to this new environment they're in. And then we get to a point where population sizes are so small and genetic drift is so strong, natural selection is so inefficient, that it basically becomes just very hard to adapt to different kinds of environments. And so this is the place where the range edge forms. So the main question here now becomes, do we actually see this in empirical data? Like, to what extent is this actually true across different kinds of species? And so this is definitely something we wanted to look at from a data synthesis perspective. And I recently tested this question in a paper that's still a preprint right now, using an expanded mammal database. So we can see these are the new locations we have. So it's a little over 1,000 sites with nearly 60 species in over 37,000 individuals. And from each of these locations, we've measured 4 different metrics of genetic composition. So the first one we looked at was a contemporary effective population size, which tells us about the relative strength of genetic drift acting on populations. 2 different measures of genetic diversity. These were gene diversity and allelic richness. And finally, for each population, we have also estimated a population-specific FST, which is a measure of genetic differentiation. And so I'll walk you through this plot I'm about to show you right now. But for each of the locations that I've showed you, we essentially measured the distance from that spot to the nearest edge of a species range. And then we use this as a predictor in a series of Bayesian hierarchical models, trying to look at this relationship between distance to range edge and genetic composition. And so we've got the 4 response variables in the models on the y-axis here. The red dashed line in the middle is the 0 line. So if our credible intervals are crossing this line, there's probably not really a big effect going on. And here are the results for effective population size. So the open circle represents our overall coefficient estimate across all of the species in the data set. It's flanked by 90 and 95% credible intervals. And then all the bubbles behind it are species-specific slope estimates. So here we've used random slopes and intercepts to account for different average levels of genetic diversity across species. And we've also allowed this relationship between distance to the range edge and genetic composition to vary for each of our species. And so what we can see here for effective population size is that overall we've got a positive effect size, so that means effective size is increasing the further away we get from a range edge, so it's higher towards the core of a range. The same pattern for gene diversity and allelic richness. And we see the opposite pattern for FST, which suggests that populations closer to the range edge are more differentiated from populations at the core. And so overall, this does support our hypothesis that there's relatively stronger genetic drift the closer we are to the edges of a species range, which could make it harder for species to adapt to new environments, and this is what leads to a finite range or a finite distribution for species. And so if we come back to our first question, thinking about biogeographic regions, we were just talking about individual species ranges, but we can really kind of consider biogeographic regions as simply layering different species ranges on top of each other. And so the transitional zone between different biogeographic regions is an area where we tend to see a higher concentration of species range edges. And so we can imagine that we might see the same kinds of forces at play here. At the transitional zones between biogeographic regions could be places where population sizes are small, populations are isolated, genetic drift is strong, and so it's again this kind of inefficient selection and very strong drift that's stopping species from really expanding their ranges into neighboring regions. And so we looked at this question as well by combining a couple of different independent data sets. So first, we used estimates of population differentiation and effective population size from my MPOP database just for the US and Canada. And so we're still talking about only mammals here. And then we also grabbed estimates of genetic diversity, specifically gene diversity, from the MacroPopGen database, which is another compilation of genetic diversity estimates from microsatellite data. The main difference here being that these were collected from literature-based estimates. And so there's, kind of a trade-off between these two approaches. Here, there's no access to raw data, but we do have a lot more species. So we use this data set to be able to ask a more comprehensive question about genetic diversity. And finally, we also brought in another database, which is tetradensity, which collates population density data from tetrapods across the world, and we grabbed these for mammals across the Americas, predicting that population density would be correlated with population size. And then here we're looking at the same kind of analysis. So again, we've used our Bayesian hierarchical models, and this time, instead of measuring the distance to the edges of individual species ranges, we're now measuring the distance to the nearest edge of a biogeographic region. And our results for effective population size and genetic diversity, we can see that these are both quite strongly positive. So we're increasing the further away we get from edges, so higher towards the core. For genetic differentiation, we are again tending to see a negative relationship with this distance to edge, which suggests that populations closer to the transitional region between biogeographic regions tend to be more differentiated from those at the core. And for population density, we really didn't see any strong effects. So we can see here the overall effect size is quite small, and the individual slopes for all of our species are really widely spread across zero, which suggests there's no general pattern going on here. And so together, these results also suggest that the global organization of biodiversity into these biogeographic regions could at least be in part due to population-level microevolutionary processes that are leading to stronger genetic drift and inefficient adaptation, which is kind of making it difficult for species to move across these biogeographic regions, therefore maintaining the kind of boundaries between these regions. This, of course, suggests a lot of interesting future questions, I think, about what's going on in these transitional boundaries, what kind of environments are here, because clearly they seem to be kinds of environments that are limiting population sizes across hundreds of species at the same time. And interestingly, I'll point out in this plot, in general, we can see from the species-specific effect size bubbles behind our overall effects here, huge majority of the species are tending to vary in the same direction as the overall effect. So this suggests that this pattern is quite consistent across all the species. And this was even true for species whose ranges overlap biogeographic regions. So, the regions are kind of the boundaries are drawn in an average place where we see high species turnover, but even for species whose ranges cross different boundaries, they also tended to have lower genetic diversity at these regions, suggesting there is potentially something very interesting happening in the environments there. Okay, and so in this next part of the talk, we're going to switch focus a tiny bit just in terms of the types of environments we're thinking about, but all the processes are really going to remain the same. And so here we'll think a little bit about how human-driven environmental change in changing the connectivity and sizes of populations could have potential for kind of reorganizing biodiversity. And so the thing here is we want to understand the extent to which different types of environmental change, and here specifically we're talking about urbanization, how consistent the effects of urbanization and this type of land use change are across species. And in general, there's a quite strong hypothesis for the types of things we expect to be happening in terms of evolution across an urbanization gradient. That's because along an urbanization gradient, as I've depicted here, we're going from these kind of large, well-connected populations living in intact habitat to ones that are becoming increasingly smaller and more fragmented. So along an urbanization gradient, we're experiencing habitat loss and fragmentation, which we then expect, of course, to affect still thinking about this kind of availability and distribution of resources, right? So along this urbanization gradient, we would expect to see population sizes becoming increasingly smaller, increasingly more isolated, and so of course drift is going to get stronger, effective population sizes decrease, that erodes genetic diversity faster, and we are expecting to see increased differentiation, particularly because in cities, we often end up with just little patches of green spaces that are kind of scattered around that can be very difficult for wildlife to move across. And so our main interest here, as I said, was trying to see how consistent this idea is across many different kinds of species. And so we tested this hypothesis in 3 different taxa, so mammals, birds, and amphibians I'll talk to you about now. And I won't show you the direct plots, but just kind of summarize in general what we found. So for each of the taxonomic groups, we did see different patterns. In general, for mammals, all of the species in the data set were negatively affected by urbanization. So for all of the species we saw in areas that had that were more densely occupied by humans, land is more transformed, genetic diversity is decreasing. For birds, we actually didn't see an overall effect across the different species in our data sets, but upon looking at the species-specific effects, we saw this was due to some species having higher genetic diversity in cities and some species having lower genetic diversity in species. So it was really more, it seemed to be a more kind of species-specific response than what we saw in mammals. And then for amphibians, we saw absolutely nothing. So there was no effect across all the species, there was no effect inside species, and the way we thought about this in the end is that, amphibians are very sensitive to their environmental conditions, they have a lot of very strict environmental requirements, and so it could be the case that just local populations in cities are being completely extirpated before we even have a chance to detect changes in genetic diversity. And so while we didn't see consistent effects across all these different taxonomic groups, I think it's quite clear that urbanization is reshaping patterns of genetic diversity, at least for mammals and some kinds of birds. That does, of course, leading from what I've talked about previously, can potentially be reshaping patterns of biodiversity at broader scales. And so when we think about urbanization, it tends to be a local process. We're talking about one city or another. But when we want to think about consistent effects across cities and species, we have to remember cities are kind of not isolated environments, right? Cities are popping up in all types of places. They're a very rapidly expanding environment. This is where most humans, the global human population, lives in cities. And so the kind of distribution of cities around species ranges is now introducing new environmental gradients that's, again, affecting the kind of existing patterns of population size and connectivity that has the potential to then kind of mess with species range edges, potentially leading to range collapses, which, of course, then has implications for the organization of biodiversity in general. And so, now we've thought about kind of the general patterns of genetic diversity. We've thought about how human environmental perturbation could potentially be reshaping these patterns of biodiversity across scales. And in the last part of the talk, we'll go into conservation to try to think about what we can do about the mess we're making. So in thinking about, specifically thinking across different scales of biodiversity, I think when we start in macrogenetics, we're thinking about population-level evolutionary processes. We're thinking about implications across the entire biodiversity hierarchy. So potentially, we have a chance here to kind of think about how we can take effective conservation strategies that can protect multiple facets of biodiversity at once. So rather than just protecting kind of existing patterns of species richness, for example, we can also start to consider the evolutionary processes sustaining this species richness to then try to conserve that as well. So to kind of, kill 2 birds with 1 stone, or conserve 2 birds with 1 stone. And so just to give an example of some of the kinds of things we're using macrogenetic data to assess in conservation. I worked on a project about looking at how well protected areas capture genetic diversity across the world, and this was in collaboration with the Conservation Genetics Working Group that was funded by the Powell Center for Data Analysis and Synthesis. And here, we wanted to focus particularly on protected areas because this is really a mainstay of conservation policy. This is something that's becoming increasingly important, especially with the agreement by a lot of parties around the world to the 30 by 30 initiative, which aims to protect 30% of lands and seas by the year 2030. And so protected areas have always been important, but they're potentially becoming even more important now, and especially because they could be a very efficient way to protect species in situ, so that's protecting them in their native habitats. And protected areas are set up for a myriad of different reasons. Sometimes it could be species richness, it could be because of cultural significance, it could be because it's a pretty landscape that humans like. But very rarely is a protected area set up to protect genetic diversity. And so we kind of went into this not necessarily expecting we might see high genetic diversity, but there is also, if we consider that all facets of biodiversity will vary in similar ways, if we're protecting areas with high species richness, if we're prioritizing areas with high species richness, what does that mean for genetic diversity? And so to just address this basic question of does the current protected area network across both marine and terrestrial realms overlap with genetically distinct or genetically diverse populations? We brought together the mammal data set that you will probably be familiar with by now, as well as a data set on marine fish genetic diversity that was compiled by Eliana Carajaliu, who is a graduate student at the University of Manitoba currently. And then we essentially just overlaid the protected area network on top of each of these locations, and we've called protected area status as essentially whether a site is located inside or outside of a protected area, and we also measured the distance to the nearest protected area. And so we're looking at the same kind of plot here. The only difference is that instead of our species-specific slope bubbles, we now just have a density distribution of the slopes. And what we can see for effective population size for both this status in or out of protected areas and the distance to protected areas, there's a whole lot of nothing going on. So there's just nothing across the board. So for all the different metrics we look at, we found that really genetic diversity, genetic differentiation don't appear to be related at all to the proximity or being located inside a protected area. And this was for the terrestrial mammal data set. When we look now turning to the marine fish, we see the same thing. So overall, these results are not surprising. So like I said, it doesn't appear that the current protected area network is overlapping with particularly diverse or distinct populations. But as I said, for the most part, these areas are not set up for genetic diversity. However, there are also different things to consider. So because there are a lot of different reasons that protected areas have been established, some of the reason why we see this kind of overall effect size of 0 is that in some cases, protected areas do capture very genetically diverse populations. So as an example, in our data set, we have a protected area that was set up for mangroves that happens to capture a very diverse population of a species associated with mangroves, which is super nice. But on the other hand, we also have data sets where a protected area was set up to protect a specific population that's extremely endangered and therefore quite genetically depauperate. And so overall, those could also be some reasons why we don't really see these relationships with genetic diversity. But I also want to venture to suggest that this isn't necessarily a bad thing. At least right now, if we aren't seeing very strong differences across protected and non-protected areas, that could mean that populations outside of protected areas have not started drastically declining. So maybe some bright news at the end of this. But these are just part of the different things we should consider if we really want to think about kind of getting the most out of our conservation efforts in trying to protect biodiversity in all of its forms. So just to wrap up here, I hope I've managed to communicate to you the potential importance and great interest of population-level processes and their relationships to broader biodiversity patterns. And just to close with saying, I think it's been a really fun and kind of interesting approach to take these complementary ways of inference from macroecology and macrogenetics, and I think moving forward it could be a really powerful way to get a much better understanding and comprehensive view of biodiversity as a whole. So with that, I want to thank all of my really wonderful collaborators and working groups that have been working with me over many years, especially in building this massive data set. I also, again, want to thank the SSE and the ASN. And finally, to everyone who has ever posted their data online, you're the best. And if anyone is interested in doing work on macrogenetics, please feel free to come talk to me. Also to my PhD advisor, Colin Garraway, we would be really happy to chat. So thank you.
Go ahead and come up to the mic if you have a couple questions. We have 2 minutes, I think, for questions. Go ahead.
Hi, Chloe. Gorgeous work.
Thank you.
And also very momentous because in this very conference center, 1.5 years, it was approved that genetic diversity will be included in global biodiversity frameworks. So kudos to you.
First time, yeah.
My question is about the time components of your data set, right? You show that with environments that are more like edgy, genetic diversity is lower, and presumably most of this data comes from the last 30 years. We know that in the last 100 years, climate change has changed exponentially. Mm-hmm. And I'm wondering if there's already a temporal signal in these beautiful data sets, or when should we expect it, if not?
Yeah, so we have collected data, mostly from relatively recent populations. I can't really speak specifically to when we should expect temporal patterns. So I will say in these data, we've obviously found signatures of effects of urbanization, which is relatively recent. In other work, I've also looked at the effects of redlining in the US, which was only practiced from between the 1930s and 1960s, so relatively recent in terms of the timescales we're thinking about. We detected effects there. They're relatively small. But yeah, I think with this amount of data, we're able to kind of maybe detect the very recent changes. But in terms of species ranges, I mean, there are other constraints of thinking about ranges are changing all the time. The ranges in IUCN aren't necessarily the kind of end-all be-all of a range, and they're very dynamic things. And so we've captured them at this one time point.
Maybe climate anomalies, right? If you have maps of recent climate anomalies or like heat waves that gets some over like a decade or something?
Yeah, we haven't looked into that type of thing, but it's something, yeah, definitely putting the data to later on.
Find Chloe later for more.
Great. So next up is Kyle David. He got his bachelor's at the University of Miami, a PhD at Auburn, and is currently, I think, still a postdoc at Vanderbilt?
Yep.
Okay. And again, from the committee, Kyle's work is highly novel, spans computational and laboratory techniques, field collections across a broad taxonomic range, and phylogenetic comparative methods. His work on duplications and the circumstances under which they have been advantageous, as well as on uneven genomic sampling across taxa, has major implications for a range of researchers. So let's congratulate Kyle. I don't know where you can put it. I can hit it. Don't forget it.
Yeah, thank you so much. I feel very privileged to be here talking to you guys. And so I appreciate you sticking with me through my impossibly vague talk title of the ecology and evolution of novelty. So like a lot of macroevolutionary biologists in the audience, I'm very interested in the relationship between genotype and phenotype, And like a lot of macroecologists in the audience, I'm also interested in the relationship between phenotype and environment. So like a lot of you, I see my work as moving across this sort of spectrum. And today, specifically, I'm going to be talking about 3 projects looking at novelty. Again, this term is extremely vague. It can mean basically whatever you want. But to me, genetic novelty means gene and genome duplications with a little bit of HDT in there as well. Phenotypic novelty, novel traits and adaptations, and environmental novelty, range expansions, or resistance to broad-scale environmental changes. So first, I'm going to talk about some polyploid biogeography work. So that's sort of the effect of environmental novelty on genetic novelty. Then we're going to be looking at some macroecological questions with niche breadth and geographic range. And finally, some work that we just put out as a preprint I'm very excited about, looking specifically about the convergent evolution of these sort of gene duplication, gene family expansion mechanisms. So to start us off, I'm going to be talking about polyploidy. So to begin, we have to start with whole genome duplication events. As I'm sure many of you in the audience know, in animals, I'm mostly, I don't do a lot of work with plants, unfortunately, but in vertebrates, we have some very prolific, well-known whole genome duplication events at some very suspiciously important junctures in the evolution of vertebrates. Whole genome duplications often precede evolutionary novelties or big adaptive radiation events. But we have to reconcile this fact with the fact that in animals, polyploidy is very uncommon. So it has this great outsized legacy in the evolution of animals, invertebrates in particular, but we have to reconcile that with the fact that it does not appear to be a winning strategy in contemporary species in the vast majority of cases. And this actually isn't very surprising. So we have a polyploid frog and a diploid frog here. Polyploids are expected to be outcompeted by diploids, especially neopolyploids. They have issues with fitness and reproductive health and survivability and all these things, where they're expected to be quickly outcompeted and snuffed out by the progenitor diploid population. However, if a polyploid emerges at the edge of a species range in sort of these more marginal habitats, we might expect that they might do particularly well. There's lots going on here, but the general idea is the expanded genomic toolkit of the whole genome duplication provides lots of raw material for natural selection to act on, and so the polyploids might be able to break out and move into these novel environments. And we do have lots of evidence for this, looking at various environmental factors, but most of this work has been done in plants. And so nobody needs convincing that polyploidy is important in plant evolution. I wanted to take a look at animals and see if we saw any similar patterns. So what first got me started was this publication in the Journal of Biogeography looking at polyploid and diploid frogs in South America. So there are actually 5 different genera represented here, all overlaid on top of one another. And so no matter what genus you belong to, it really seems like there's this polyploid-specific habitat in the southeastern region, almost to the exclusion of diploids. So I thought this pattern was really fascinating. To me, it speaks to like a specific environment that selects for polyploidy, which is really interesting. When we do some phylogenetic comparative methods, the variable that really pops out as being significant is temperature seasonality. So this is the standard variation of temperature annually. Okay, so great, awesome. Maybe temperature variation represents this like challenging environmental factor that the polyploids are better adapted to. But there's an alternative explanation in that temperature shocks are a common way to induce polyploidy, especially in like aquaculture. This is temperature, heat and cold shocks are known to induce the sorts of meiotic errors that produce polyploids. So maybe we're not selecting for polyploids, they're not adapting to this environment, they're just formed here at a greater incidence. So to expand this work a little bit, I'm only working with about 8 polyploid species here. I'm now going to look at across all amphibia, as well as ray fin fish and insects. So here are the polyploid species I was able to identify in the literature. And here is a global map of polyploid frequency in these 3 clades. And I don't know if any patterns jump out to any of you, but it turns out there is a latitudinal gradient here. We just heard about a different latitudinal gradient in the last talk, but here we find that polyploidy increases with distance from the equator. So again, very striking pattern mapped out, but now we have to go in and see, what the heck is going on? What do we think is driving these trends? So I assembled a bunch of different environmental variables and saw which were significantly different between polyploids and diploids. So at the top, you can see, for example, polyploids are associated with more seasonal environments. Not surprising, given what we just learned about the South American frogs. All the way at the bottom, polyploids are also associated with colder environments, so a negative relationship with temperature. Again, not surprising, given this latitudinal gradient where they're more common at higher latitudes. When we do some relative importance analysis, however, we find that glaciation really seems to be the underlying factor driving this latitudinal trend. So I thought that was very interesting. To me, it speaks to a model of polyploid evolution where you have a glaciated Earth. We have the glaciers retreating. This is a new environment. And maybe the polyploids are better adapted, right? They have these expanded genomic toolkits. They have maybe neofunctionalization or just the greater mutation rate where they can get these 1 in a million mutations that helps them break into a new environment. I've also, this work has been done in plants as well. I also looked at invasiveness briefly and found that in amphibians and ray fin fish, polyploids are more likely to be invasive. Again, that speaks to sort of their adaptive potential being introduced into a radically different environment and being able to become established and persist in a new novel environment. So that concludes this chapter. I'm reporting the existence of a latitudinal polyploid gradient in 3 animal clades, complementing work in plants that has also shown similar patterns. Unlike in plants, I attribute this mostly to glaciation cycles. This provides support for the hypothesis that polyploids are maybe better suited to newer or more extreme environmental conditions. All right, so that's the first bit down. Now I'm going to look at the relationship between phenotype and environmental, phenotypic novelty and environmental novelty, looking at some relationships between niche breadth and geographic range. So let's bring up some frogs, of course, again, even though this work is actually not on frogs. We have a specialist species in green that has a relatively restricted niche breadth, and our blue species is more of a generalist. We might then expect a common hypothesis in macroecology is that this is reflected in their geographic range. If you're a specialist, you have a restricted range compared to a generalist species. However, this is in practice kind of difficult to test because the first thing we need to do is figure out what the hell we mean by niche breadth. Are we talking about diet, habitat, climate resilience? And the second issue is often we define niche breadth and geographic range from locality data, from these point estimates of where species occur. However, if there is, in whatever variable we're testing our niche breadth, if that has spatial structure to it, these factors become autocorrelated and you can never tease apart what you're actually talking about. You're always going to get a correlation between niche breadth and geographic range. So to get around this, unfortunately, I had to leave frogs behind. And this is now in my postdoc working in a big comparative data set of Saccharomycotina yeasts. So we have a little over 1,000 species here, and each one has been experimentally validated on 17 different carbon substrates. So this is really helpful for a couple of reasons. One, the feeding mode of yeast, right? They have this external feeding mode. They kind of live on the substrate that they eat. So this is a nice way to look at like diet and habitat condensed into one variable. And this is also experimentally validated, so totally removed from where the yeasts are actually in the environment to sort of tease apart these two factors. It's an independent way to measure niche breadth. So again, for example, our purple species all the way on the right, that would be a specialist because it can only grow on one of these substrates. So this is all thanks to Dr. Abby Labella and Dana Opulente as part of some awesome work that all was done over many years before I got to the lab, and I just got to swoop in at the last minute and like do a bunch of fun analysis. Another really cool thing from this paper that they put out is they found that the genome size, or the number of genes, also corresponds with this niche breadth. So If you're a generalist, you're more likely to have more gene copies, so this is also a way I can kind of get at that earlier idea of polyploidy indirectly, where I'm also sort of testing this underlying trend that, restricted ranges are more likely to be specialists, and specialists are more likely to have smaller genomes. Maybe, we'll see. And so to test this, I did some machine learning species distribution modeling to infer species ranges for 186 species that I had sufficient data for and that the models performed well at. I'm happy to get into the nitty-gritty with anybody later, but overall I was pretty happy with these models. And then when I stack all 186 species on top of each other, I get this distribution of global yeast diversity. And so now I'm able to compare all species to just our carbon generalists, or specialists rather, sorry. And when we do this, we do indeed see that specialists have restricted geographic ranges. You can see here, if I order it by range size, all our specialist species are more likely to have smaller ranges than species that can grow on a wider number of carbon sources. So how are niche breadth and geographic range related? I inferred the species distribution maps for 186 yeast species that all have like very nice genomes and niche breadth data thanks to my collaborators. I did find that metabolic breadth in these species, carbon breadth, correlates with geographic range size. Because we know specialists also have smaller genomes, this provides indirect support for that idea that additional genes may infer environmental plasticity and resilience to more challenging environments. So I hope everybody is with me so far. We're now going to move on to the last work that I'm very excited about, looking at the mechanisms by which these larger scale macroecological patterns may have occurred by looking at these gene family expansion events. So I'm going back to my yeasts using a modified data set with help from my lab mate, Mary Claire Harrison. We're losing a couple species to get a lot more substrates. And again, these are various chemical substrates that have been tested in the lab for any given species that do or do not grow. So let's say we have a distribution that looks something like this, where we have these species that are able to grow on these traits. And again, now I'm digging into the genotype a little bit. We can see the gene count. One might infer that this duplication, this additional gene, has granted the ability to metabolize this particular carbon source. And there are lots of famous cases of this where either gene or genome duplications have been attributed to these novel traits and adaptations. But as we are all very good evolutionary biologists, we do our comparative phylogenetic methods. If we have our tree that looks like this, and our trait was gained here, and our gene duplication occurred here, that actually leaves us with no degrees of freedom to make meaningful statistical correlations, right? This is a very common issue known as, Darwin's scenario. He himself commented on this, and it's been shown that this shows up in our phylogenetic comparative methods as well, where you cannot tease apart these ancestral single events. And so that's why a lot of these cases now have big asterisks next to them, that the specific timing and stuff is recently drawn some controversy in some cases. So one way around this is, one of the great things about this yeast data set is these traits have all evolved many different times. So in the smaller case, we have erythritol evolving 66 times versus lysine, which evolved thousands of times. So each one of these traits evolve dozens to hundreds, or in some extreme cases, thousands of times. This provides a lot more statistical support to look at these questions of convergence and the underlying genetic mechanisms that produce these novel traits. So let's say we have our phenotype and genotype here. So here's an example, right, of a very famous example of a convergent trait. We have camera eyes in vertebrates and mollusks, as well as compound eyes in dragonflies. And very famously, these are all the result of the same underlying genetic, this master controlled gene, PAX6. The homolog is found in all these different cases. So I would call this, please don't get mad at me, I would call this parallel convergent evolution for the purposes of this talk. These are the definitions I'm going with, where you have convergent phenotypic evolution affected by the same underlying genetic elements. Conversely, we have another famous example of convergence looking at these antifreeze proteins that show up in many different fishes, both North and South Pole. And even, right, this is AFP1, this is the same family, but it was found that they result from many different underlying genetic elements. So I would call this, for the purpose of this talk, I would call this a non-parallel convergent evolution, where you have convergent evolution that is not the result of the same genetic elements. So my fundamental unit of analysis to this problem should be familiar to a lot of you, looking at phylogenetic logistic regressions, where for any given gene family, I can see how big this gene family is in species that do or do not grow on a given substrate. And I've modified this slightly, borrowing some tips from some GWAS analysis with help from my collaborators Matt Powell and Josh Schraber at USC. Again, happy to get into the nitty-gritty with you guys later, but this is designed to, again, get around this idea of Darwin's scenario where you are trying to increase our statistical support to avoid these spurious correlations from these single events. Okay, so I do all that for all my gene families and all my traits. And the gene families that turn out to be significant are enriched for these gene ontology terms. This is a nice gut check for me. It's nice that I'm looking at metabolic traits, and it seems like the gene families that are significant are involved in metabolism. So that's awesome. So here's one example of this type of analysis, looking at one trait gene family correlation. And hopefully, it's not a stretch to see that the larger your SUC2 family is, the more likely it is you're going to be able to metabolize raffinose, which to me speaks to a sort of convergent mode of gene family expansion that occurred multiple times in the evolution of this trait. And if we map on our hypothesis of trait evolution and our inferred gene family expansion events, gene duplications and horizontal gene transfers, yes, this looks pretty good to me. Again, I don't think it's a huge leap in logic to say that these gene family expansions correlate or precede trait gains in this system. And so I can do this not just for raffinose, but for every trait. I can look across all the gene families and see, do any of them show these signatures of parallel convergent evolution? And yes, we do. We have parallel evolution in 80% of our metabolic traits tested, where we have at least one family that shows these signatures, where you have the same gene families being involved multiple times in the evolution of a given trait. So if you'll sit with me for another tortured metaphor, if we have this swan phenotype evolving independently, rather than developing new ways to produce this phenotype, it seems like we are having the same gene families are repeatedly undergoing gene transfers and duplications. It's the same gene families, the same players involved in the evolution of these traits, some of which have shown up, hundreds of times across the evolution of Saccharomycotina yeasts. Something keen-eyed audience members might also have noticed is our traits are hierarchically clustered by which gene families, the strength of the effect of across all gene families. So if you're closer together, you have the same gene families are important or show signatures of convergence in the evolution of this trait. So we see lots of these butane protein or structural isomers. We have nitrate, nitrite. What's my other example? Xylose and xylitol. One is a derivative of the other. We have this nice clade of glycosides here. It seems like there is the chemically similar elements are using the same gene families. So to me, this invites the comparison. Instead of looking at each trait, now let's look at each gene family, and are some gene families contributing to the evolution of multiple traits, not just one. So when we do this, we do find some exciting results. So all the way on the left, the vast majority of gene families are not involved with any trait. That's okay, right? This is across the whole genome. We wouldn't expect most genes to be, metabolically relevant for a given trait. The majority of those that are significant, we do see convergent signatures with just one trait, so it is this sort of one-to-one relationship. But we do find a minority moving up all the way to 13 different traits were significantly targeted with just one gene family. So I thought this was pretty exciting. This ends up being a alpha glucoside transporter in Saccharomyces. I think it's called MAL31 or MAL11. There's a couple in the ortho group that I'm looking at. So this is a known maltose transporter. So here we have our graph again. Hopefully you can see that the larger your AGT family is, the more likely it is you're going to be able to metabolize maltose. But not only that, but if we overlay all 56 substrates, you hopefully see a trend as well, where this gene family seems to not just contribute to maltose metabolism, but a wide variety of different carbon substrates. So this seems to be what we're calling in the preprint a keystone gene family that seems to control a wide variety of different metabolic processes, again, in parallel over the 400 million year evolution of the Saccharomycotina yeasts. So to bring back my little origami figures, not only are we finding the same gene families over and over again in the evolution of convergent traits, but a select few of these gene families seem to be able to contribute to multiple different phenotypes as well. So, do gene families expansions produce novel traits? I would argue they do. I find parallel associations between gene family size and trait presence and absence in 80% of cases. This was very surprising to us. It seems like parallel evolution across, again, these really deep timescales may be more common than previously believed, at least in yeasts. We also find these sort of keystone gene families that contribute to convergent evolution of multiple different traits. And altogether, this does support my hypothesis that these gene family expansion events do lead to the evolution of novel traits in metabolic breadth. So, if we take our significant findings from each of these three chapters and we rearrange them a little bit and we squint really hard, we might see a overall macroecological evolutionary framework about how these gene family expansions lead to novel traits. These novel traits lead to expanded gene family sizes, or these gene family sizes lead to novel traits, which lead to expanded range sizes, and maybe that's why we see more polyploids at these more extreme new environments like introduced invasive species or deglaciated areas. So that's all I have for you today. Great timing, I guess. I'm happy to take any questions if there's time. Yeah, this was just out this week, this blue one here. So speak now or forever hold your peace. If you really didn't like what I said about it, please let us know, because it's going to go into review pretty soon, hopefully. All right, thank you very much.
Great talk, Kyle. Beautiful slides as well. Really amazing.
Thank you.
I'm just wondering with the polyploidy, with your first study there, with most polyploid vertebrates, they're usually parthenogenetic. So I'm wondering whether you looked at like correlations between the polyploidy and an asexual reproductive mode. I mean, perhaps not in the frogs; they're not parthenogenetic.
Right. Yeah. So
for any of the others. Just because geographic parthenogenesis is usually, parthenogens tend to be more in regions that are harsh, or I don't know. Just wonder whether there's any correlations there.
yeah, that is not something we looked at. I'd be amazed if that wasn't something going on. Yeah, we just, it's well established in you know, many arthropod cases, but sexual mode is either not reported in a lot of the ones that I was looking at, but absolutely picking like, I don't know, I'd love to look at like Daphnia or Artemia or you know, one of these arthropod groups and investigate this, because I am sure that is a relevant factor as well.
Yeah. Awesome, thank you.
All right, all good? All right, thank you guys so much.
Bienvenidos, and welcome to the last 3 of the 5 American Society of Naturalists Early Career Investigator Awards. My name is Chris Moore, and I'm one of the 4 members that selected these 5 awardees. Just to give you a quick background on the Early Career Investigator Award, it was established in 1984 to recognize the outstanding and promising work by investigators who received their doctorates in the 3 years preceding the application deadline, who are in their final year of graduate school. The committee is composed of me, Martha Munoz, Jean-Philippe Guibert, and Chair Eleanor Caves. So this was such an honor to serve on this committee, and although we wish we could give awards to everybody, we ended up finding 5 just wonderful, inspiring people with wonderful and inspiring work. So I first would like to introduce Stepfanie Aguillon, who is at UCLA. And Stepfanie's work seeks to understand how and why reproductive isolation develops, which is key to understanding the diversity of life and how it evolves. She elegantly uses approaches across levels of organization, from individuals to species, leveraging genomics, field collections, museum specimens, and lab experiments. So with that, we would like to present Stepfanie with the award. If we could have a round of applause. And I'll welcome Stepfanie up to the stage.
It's really cool to get a plaque. I didn't know that was going to happen, so it's really exciting. Well, I'm really excited to be here and really honored to be one of the recipients of this award. I think also this is going to be a really fun session with 3 ornithologists. I'm going to be talking about fish, not birds, but yeah, very excited to be here. And let me see if the clicker works. It does. So I wanted to start with my acknowledgments to thank the many people and organizations that have made the work that I'm going to tell you about today possible. In particular, Renee and Jeremy are here, so thank you very much for all of your impacts on my science, but also my career. And with that, let me make sure the time's going here. Okay. I will go ahead and get started. So my research and my passion for biology really starts from looking out in the natural world, and I think that's probably something shared with many of you in the audience. I'm really fascinated by the diversity of shapes and sizes and colors and behaviors that are present in nature. And this is something that has really influenced and shaped the way I think about the science that I do. And for me in particular, when I see these sort of biodiversity slides, is I key in on these organisms that either look sort of similar, you think they maybe act sort of similar, and I want to understand why that is. And that's sort of the basis of what my research program does. So I think about the mechanisms that lead to and maintain species barriers, thinking about why organisms look the way they do, why they act the way they do, what keeps them separate, what brings them together. And this is what I'm going to be talking about today. So I've decided with my talk here to focus in on one sort of expansive project that was the basis of my postdoc. I didn't say this at the beginning, but I'll say again at the end that I just started my lab 28 days ago. So I've been an assistant professor for less than a month. And so I'm going to tell you about the work that I've been doing for my postdoc primarily. And so I'm going to build a roadmap here of different barriers and mechanisms that we can think about in the speciation process. So we often think about things as being either pre-mating or post-mating barriers. And the point here is that they function either to prevent mating from happening, or once mating has happened, they influenced the outcome from hybrids or offspring that are produced. So for pre-mating barriers, you can think of things like ecology. So perhaps if an organism prefers a type of flower, that flower distribution is going to determine who that organism overlaps with. You might have differences in behavior, so song, for instance, in birds. If a male sings a particular song, females might prefer that song and choose to mate with individuals that sing the right song. You might have simple mechanical differences, so if individuals just can't mate with each other, mating doesn't happen, like the unicorn and the dragon, obviously. So these are a whole host of pre-mating barriers, but there are also post-mating barriers. And these are things that are due to genetic incompatibilities that influence things like hybrid inviability, hybrid sterility, basically cause a bad time for your hybrids. And there we go. Okay. So if you're thinking about these species barriers, you might be thinking about when these species barriers break down. And you might be familiar with this example of hybridization when you have a horse and a donkey mating. This produces the mule, and from this example, you might be really familiar with these mating barriers, where we have these post-mating barriers that are very strong. So mules, as you might know, really hardy pack animals, which is great if you're going down to the Grand Canyon, but don't produce any offspring. So you might think of these as an evolutionary dead end. And if this is the only example of hybridization that you're familiar with, this is something that I want you to take away from the talk. This is not actually all that typical. So in fact, hybridization is very common across the tree of life. We know this now in much more detail than we did even just a decade ago. And the great thing about knowing this now is that we know that not all hybridization events are evolutionary dead ends. In fact, hybridization can be a creative force in the evolutionary process. These examples that I'm showing you here are all organisms that have had hybridization in their evolutionary history. This has moved genes across species boundaries, and these have been involved in creating the changes that we see today. And so what I'm going to tell you about is, sorry, let me see if I can see where my next slide is so I can, there we go. Okay, so hybridization is also a really great lens to study evolution, and this is one of the primary modes of my research program. So thinking about when we have these previously separated species that come back into secondary contact, when they produce hybrids or don't produce hybrids, this gives us a lens to look at the way these organisms look, how they act, if we have mostly males produced, mostly females produced, and this gives us a really interesting window to think about the evolutionary process. So my research leverages an approach that takes a bunch of different methods to think about speciation and hybridization. So I use genomic sequencing, behavioral ecology, organismal biology, both in the lab and in the field, museum specimens, and underlying all of these approaches are hybridizing taxa. And what I'm going to tell you today is a story using this roadmap that I built at the beginning, looking at the mechanisms that lead to and maintain barriers in swordtail fish. All right, so going through a bunch of these mechanisms in depth and trying to understand what makes these species different. So swordtails are a really great system for thinking about the speciation process. So these are small freshwater fish distributed in eastern Mexico. And for me, swordtails are really exciting because there's abundant phenotypic variation. So these are a variety of males from the species in this genus. And what you can see is lots of colors, lots of shapes, lots of different patterns. They have these fin appendages. So there's a ton of phenotypic variation. But despite this, hybridization is actually really common. So these lines connecting these different lineages here are instances of hybridization that have occurred in the evolutionary history of these lineages. And a bunch of the work in hybridization, in particular in this group, has been done in the northern swordtail. This is thanks to my postdoc advisor, Molly Schumer, and her collaborators. And she's mostly worked in the species Birchmani and Malinche and their hybridization. So these are sister species. What I've been doing for my postdoc is adding in this additional species, Cortezi, and that's what I'm going to be telling you about today. So we've been working in this new hybridizing system between Xiphophorus cortezi and Xiphophorus birchmani. They overlap in these small ranges in eastern Mexico where they come into contact in these river drainages. We've previously studied 2 hybrid populations, and what I'm going to be telling you about today is an independent incidence of hybridization between these 2 species in the population Chapulhuacanido. All right, and so to tell you about this population, I'm going to outline the genetic methods that we've been using. I'm just going to go through this in just 2 slides, but I'm happy to talk about the approaches that we use in general later. So what these methods require is some kind of tissue sample, molecular lab work. In this case, we're doing low coverage whole genome sequencing. And then a lot of computer work, which is probably not what you think of when you get into biology. But what we're doing here is local ancestry inference, and this is the method that I'll tell you a little bit about now so you can understand where the numbers in some of these plots are coming from. So for local ancestry inference, we are taking pure references and using them to define ancestry informative sites. So we take population of species 1, population of species 2, and define regions of the genome that differ between them. We use these to assess individuals of unknown ancestry. So we can say, for instance, we have an individual number 1 is 100% green in their genome, or individual 2 that's 50% green, 50% blue, and so on and so forth. We can then use these colors that I've shown you basically and what I've said verbally to calculate an overall genomic ancestry. So for instance, we might have 100% species 1, 50% species 1 in the genome, et cetera. And so when we look at this Chapawacanido population, this hybrid population, we find this interesting bimodal distribution of ancestry. So we have 2 clusters of individuals. We have this blue cluster of individual that is nearly pure Birchmani in their genome. And this light green cluster of individuals that are admixed, they're about 75% Cortezi in their genome. And this bimodal distribution itself is pretty interesting. You might expect in a hybrid population to have everything sort of collapse into one. So that's sort of an intriguing thing to ID right at the beginning. Even more intriguing is that this bimodality really mirrors what we see in these independent hybrid populations, where we have this nearly pure Birchmani cluster and this additional admixed cluster. So something intriguing going on there. And even more so, this distribution of ancestry has been maintained over time. So this is what the contemporary ancestry looks like. But if we go back all the way to 2003, we see this bimodality mirrored there as well. So something has been maintaining this ancestry distribution over time. So looking at this roadmap, we've ruled out mechanical differences, unfortunately, so the unicorns don't matter. These clearly mate with each other. We have this potential role for some sort of post-mating barrier, perhaps, where we have this repeated bimodality in the ancestry clusters, so maybe something going on there. We'll explore that more at the end of the talk. Perhaps some sort of behavioral barrier, since this distribution of ancestry has been maintained for many generations. And that's what we're going to explore first. So when we think about this behavioral barrier, the first question is assortative mating happening? And if it is, what's the strength of assortative mating? So to do this, we've taken advantage of the really interesting biology of these fish. So these are live bearers, which means they are inseminated internally. They give birth to live young. And so if we collect females that are pregnant in the wild, we can bring them back into the lab and dissect out their embryos. And we can sequence the mother and the embryo and compare their ancestry. When we do that, if the mother and the embryo ancestry are really similar, we can infer that the male she chose to mate with was similar in ancestry to her. And this suggests really strong assortative mating. Whereas alternatively, if the mother and the embryo ancestry are really different, we can infer that the male she chose to mate with was very different in ancestry from her, which suggests weak assortative mating. So what does this look like when we look in this hybrid Chapawacanido population? We actually find really strong assortative mating. So I'll walk you through this plot and sort of make it a little nicer for the viewer. So we have these 2 clusters of ancestry, this admixed cluster of females and this Birchmani cluster of females. The x-axis here is the important one. This is the difference between the mother and the embryo ancestry. The farther away you get from the 0 line, the more different the mother and the embryo are, so the less assortative mating. Because that's the important axis, I'm going to cut away the middle there. When you spread out the points, hopefully you can see most of them are clustered on the 0 line, which suggests very few differences between the mother and the embryo. Which suggests this strong assortative mating. And in fact, when we do simulations that we parameterize with population data, we see that 100% assortative mating simulations fit this data best, suggesting really strong assortative mating here. And this is a really stark pattern when you compare to simulations of random mating, which have many, many points far away from the 0 line, which is just not what we observe in in the population. So going back to this roadmap, so we have evidence here now of some sort of behavioral barrier. And what we've been doing after we figured that out is experiments to try to understand if female mating preferences are consistent with this assortative mating, if they sort of explain this pattern. So we've been doing experiments, and as you can see, the males of these species have very different visual phenotypes. They also have very different pheromones. And we've been doing experiments to try to tease apart if females are using these as cues for mating. So we have trials where we can introduce a female into a tank with males on either end. If we're testing visual cues, we can use just a clear barrier separating the female from the males. We've also done a set of experiments with visual and olfactory cues, where this clear barrier instead has holes, allowing water flow in between. When we do these experiments, we find that female preferences are complex. So, and if you've ever done behavior work, this probably is not a surprising result to you. Complex. So strength of preference is on the y-axis here, with positive values indicating preference for Cortezi males, negative values preference for Birchmani males, 0 is no preference. In these visual experiments, we see Cortezi females strongly prefer Cortezi male cues, no preference in Birchmani. When we add in olfactory cues, this preference goes away for both, so no preference there. We've, alongside these experiments, been trying to dissect apart the isolated role of the male pheromones in the laboratory. And we've been doing this by introducing just the pheromone cue into the sides of tanks with females in them, allowing females to move around and seeing where they spend their time. We record videos and use tracking software, which is really helpful. So we have these cue zones on either end, a neutral zone in the middle. We introduce these females, and this gives us these nice heat maps from this software to see where they spent their time. And this is work that's been done with Sophia Hazy-Cox, who's an undergrad here, former undergrad, I guess, and is looking for grad school here in the fall. So if you're interested, please talk with her. We didn't find any clear female preferences, but we did find some strong behavioral differences that I want to just mention here. So in both the allopatric populations of fish and fish from this hybrid population of Chapawaka nido, fish with a lot of Cortezi ancestry in their genome, either Cortezi, pure Cortezi, or these admixed cluster, spend much less time in the neutral zone than expected by chance. So they are much more either bold or exploratory than you would expect by chance, and much more so than Birchmani. So it seems like they perhaps are using the environment in a different way, and this potentially could translate to what they're doing in the wild. But more hopefully soon. So we have some hints that female preferences are involved here. I'm putting it in small letters because just hints, and there's a lot of complexity there. And some potentials that there is some sort of ecological difference going on as well. Now we've really thoroughly explored this pre-mating side. So what we're going to do now is think about post-mating. So alongside of all of these experiments that we've been doing, we've also been conducting artificial crosses in the laboratory. And we've been doing this in both directions with female Cortezi and male Birchmani, as well as female Birchmani and male Cortezi. And when we do this, we see dysfunction in both of these crosses. So this first cross has been successful in the lab and in stock tanks, but there's a heavily skewed sex ratio in the offspring that are produced. We get about 6 males for every 1 female. And if you're in the speciation world, this might be an intriguing hint of something interesting going on. This other cross has been largely unsuccessful. So with female Birchmani in this cross, we get very few offspring. We've produced just a single F1, even using artificial insemination. But using dissections of these artificially inseminated females, we do see that fertilization is happening, which is intriguing. So looking at these mechanisms again, does seem like there's some level of hybrid inviability. As I mentioned at the beginning, genetic incompatibilities are involved in all of these here. And we have hints even more so that genetic incompatibilities are involved. So we've used the successful F1 cross to produce F2s, and when we look at the embryos that are produced from this cross, we have these 2 interesting phenotypes. We have some individuals that are developmentally typical, and other individuals that are developmentally stalled. So these are embryos dissected from the same female. They are siblings. We did this at the same time. And when I show you the ratios that we get these embryos in, you might get a hint that something is going on, especially if you think a lot about genetics. So this is really intriguing and was especially intriguing to us because some recent work in Molly's lab has focused on this genetic incompatibility between Birchmani and Malinche. And this is work led by a grad student, Ben Moran, who's on the postdoc market this year. Ben's also in the audience if you're interested in talking with him. And this is an incompatibility between the Malinche mitochondria and two nuclear-encoded genes in Birchman eye, such that when you have this bad combination, you don't make it, basically. And the reason I bring this up here is because ancient introgression has moved the mitochondria from Malinche into Cortezi, such that when we look at the diversity of mitochondria here, Cortezi mitochondrial diversity is nested within Malinche mitochondria, which is very different from the Birchman eye mitochondrial diversity. And so not only that, but the nuclear part of this incompatibility, so the Malinche alleles for both of these nuclear encoded genes, have also introgressed into Cortezi. So I'm showing you here the amino acid differences in these genes for Malinche, Cortezi, and Birchman eye, and hopefully you see that Malinche and Cortezi are the same and different from Birchman eye. So we have interesting movement of all of these components of this incompatibility, and we have some strong suspicions that this could be important in our F2 hybrids, and in particular because all of our F2 hybrids possess the Cortezi mitochondria because of the direction of the cross, which means this incompatibility could just be working in these hybrids. And that is, in fact, what we see. So when we look, for instance, at this NDUFS5 gene, what we find is that 100% of the stalled F2 embryos have 2 copies of the Birchman eye allele at this gene. So for normal embryos, they are either heterozygous or have 2 copies of the Cortezi allele. Stalled embryos, 100% have 2 copies of the Birchman eye allele. And we see a similar, though slightly different pattern with the other gene. This one we know from the previous work that it works in adults. And in adults, we see a depletion of F2s with 2 copies of the Birchman eye allele at this gene. So we have intriguing hints that this incompatibility is working here in this system as well. And because of these patterns that I just shown, we have strong selection actually against the Birchman eye allele in both of these genes. For NDUFS5, the selection coefficient is nearly 1, but also quite high for NDUFA13, which is interesting. And this strong selection leads to segregation distortion in the genome. Rather than having 50-50 ancestry, which is what you would expect in an F2 cross, near these genes you have very skewed ancestry, which is interesting. Okay, so this also adds this really interesting new complexity to our and the field's understanding of how historic gene flow can interplay with present-day reproductive isolation. So the idea that all of the components of an incompatibility in a pair of species can move into a third species and then also influence reproductive isolation is pretty intriguing, and to our knowledge, it hasn't really been described at this level before. If you have other examples, please talk to me later. But really adds this interesting layer of historical hybridization influencing reproductive isolation. So we have this interesting result here. Okay, back to the roadmap. Hopefully I've convinced you that incompatibilities are really important. If you are, and hybrid sterility is not important because we have F2s, doesn't seem like that's involved. If you are a speciation person, you might be thinking, but wait, there is another category here that you haven't talked about, which is this sort of in the middle, often cryptic area of speciation barriers, which is post-mating, pre-zygotic. These are things that happen after mating, but are often cryptic, like choice of sperm by a female that we can't really observe easily. And we've looked at these barriers as well. This has been work in collaboration with Dan Powell, who is a postdoc also in Molly's lab. He's starting his lab at LSU soon and giving a talk tomorrow, so you should check it out. He's also here. And what Dan has found is interesting morphological differences between the different species and the F1 and F2 hybrids, as well as some interesting motility differences. And so we have these hints that there's also something going on at this really interface between pre-mating and post-mating barriers. So I'll do like a slightly faded check there. This sort of interesting hints. And this is a direction I think Dan is excited to go in the future. Okay, so what have I told you about swordtails? Species barriers are multi-layered. Hopefully I've convinced you that genetic incompatibilities play a huge role, but there are other mechanisms as well, and hopefully you feel like I've thoroughly covered many of them in this talk. So I've told you a lot about my interests in speciation and hybridization. What I sort of forgot to mention at the beginning is that my lab just opened. I've been an assistant professor for like 28 days, so very new. So please get in touch if you're interested in collaborating or joining my lab. I'll leave this slide up at the end as well. I wanted briefly, while I'm here, to tell you a little bit about some of my other interests. So I told you about speciation and hybridization. My other parallel research interest is in coloration. And this is something I think that fits really well with an interest in looking at organisms and seeing why they look different. And this is something that biologists have been thinking about for a long time. And I'm, excited to continue the tradition, I guess. We're stuck. Oop, there we go. That went twice. Okay, well, so I am really interested in the evolution of color and patterning as well. Sorry, this slide's being weird. So I have done work thinking about the genetic basis of color in birds, as well as thinking about what community science sightings can tell us about color evolution. Also doing some work in swordtails as well, thinking about the genetic basis of patterning. And this is a direction I'm really interested in going in. And I wanted to highlight on this slide these birds, because actually I'm not a fish biologist, I mostly am a bird biologist. And I've been working in flickers in particular since 2015. I felt very aged when I was looking for photos of me with flickers. You'll notice that I'm wearing the same hat and shirt in both pictures. So it's been a while, but not that long, apparently. And flickers are a system I'm really excited to continue working on in my lab. And let me tell you a little bit about them. So they have these really beautiful phenotypic differences. So I think very obviously when you look at them, you're like, wow, those are really cool colors. And they also hybridize in the Great Plains. So red-shafted flickers are distributed broadly in the west, and yellow-shafted flickers broadly in the east. And they hybridize in this orange region in the middle of North America, from northern Texas following the Rockies up into Alaska. So they have both of this hybridization component, speciation component, as well as this color component that really interest me. And so my lab's going to continue using flickers to study hybridization, thinking about epistasis between pigmentation genes, thinking about migratory differences between the two species as well as their hybrids. Flickers are sort of unusual that they migrate, which makes their hybrids and what their hybrids are doing for migration kind of intriguing. And they also have interesting transposable element and structural variation in their genomes, and trying to understand how that interfaces with hybridization is one of my goals. And so we have this hybridization aspect, but I'm also really interested in the evolution of color and using these birds to study that. So these are red-shafted and yellow-shafted. They're closely related species. Their most closely related species is the gilded flicker, which actually looks like red-shafted flicker, but with yellow wings. These also hybridize with red-shafted flickers in a mosaic hybrid zone. These are Sonoran desert specialists, so there's this interesting layer there. But then I also wanted to broaden this a little bit to the genus Colaptes and more broadly, and show you some other images of birds in this genus. And what I want to highlight here is the sort of variability in patterns across the body and their color. And I've thought about these for a long time, and I really am excited to try to understand how and why they have evolved. So we have these colors and particular patches on the body. They change colors. They might be red, or they might be black, or they might be yellow. And I'm trying to understand how this comes to be. Is this a similar genetic architecture that's producing many different colors? Is this something that you can just turn on and off and mix and match in different species? Is this the result of hybridization? And so this is a big direction that my lab's going to be going in. And if this is something that excites you, please get in touch. And with that, I will leave this slide up. I think I might not have any time for questions, but I don't actually know because I'm not sure. But I will take them if I can, but I will be here for the rest of the meeting.
Okay. So thank you for coming to my talk. Variation within species is sometimes driven by phenotypic plasticity in different environments. Like, for example, in these vetch aphids, when the food source is favorable, they hatch individuals that don't have wings. But then when the plant gets densely populated, they start hatching individuals that have wings and can migrate or move to a different plant. And so in theory, this phenotypic plasticity can promote speciation because the potential to change is already present under different circumstances, right? And the change in one trait can influence changes in other types of traits that can feed each other and accelerate the speciation process. And then the genes responsible for reproductive isolation are more followers rather than initiators of the process. Migratory behavior is a behavioral syndrome that involves the correlated evolution of many different traits. This is a fantastic bird. It's the Arctic tern. They breed in the Arctic and then migrate to overwinter in the Antarctic. And you can see here tracked individuals that do this twice a year. It's a very long journey, and to accomplish this, they need different mechanisms. Like, for example, the evolution of sensory mechanisms to go in the right direction. They need morphological evolution so that they can fly efficiently. They also need physiological mechanisms to accumulate a lot of fat so they can actually fuel these long-distance migrations. And they also need sensory mechanisms to live at the right time. Migratory behavior has evolved across the avian radiation in different families. It has also evolved across different functional groups. Small birds migrate, large birds migrate. It's across all of them. But I like to stress that being non-migratory is also very extraordinary. In the tropics, non-migratory birds sometimes face very strong predation pressures, like this poor thing. And there's also sometimes very large amounts of parasites. They also face seasonality because there's precipitation seasonality in the tropics. And then I'm amazed by the birds that can, stay year-round in very cold temperatures. So there's trade-offs between these two strategies that are very different, and trade-offs that likely involve many traits that switching from one to the other can accelerate the speciation process. Something that I found very interesting when I started studying this behavior is that within migratory birds, there are about 900 species that also have intraspecific variation. So this is an example, the yellow warbler. In the map, what you can see in orange is the breeding distribution of migratory yellow warblers. Yellow is the areas where they're passing by, blue is the areas where they overwinter, and then the arrows that I drew there are pointing to places where the yellow warbler stays the whole year. They are non-migratory. And I also like to include the pictures of these two birds. So in this case, this non-migratory yellow warbler has also evolved different coloration, and they breed in mangroves in the Caribbean. So the questions that motivated my PhD was basically if these are early stages of the speciation process, if they're speciating, and if it can promote diversification. More broadly, I would say I'm interested in whether behavior can promote species divergence. Oh no, my picture is not there. That's so sad. There's a beautiful bird there, the fork-tailed flycatcher. It has a very long tail. That's very sad. No problem. So I can show you the distribution of these birds through time. This is a video from eBird, and you can see in darker purple is areas where they are very abundant. And then you can see that there's a group of birds that migrate to South and South America, and then they come back to North and South America, and some then stay all the time in North and South America. And they co-occur during part of the year, and this is consistent throughout the years. And so through population genetic analysis and phylogenetic analysis, I found evidence that from the migratory population, there was a drop of migration that led to divergence from the parental population in birds that don't migrate. I also found that in those areas where they coexist in North and South America, migrant birds are in an arrested migratory breeding state. Migratory birds are shown here in blue. They are likely just replenishing from the long-distance migration, and we can tell breeding condition in birds because females have a brood patch and males have a cloacal protuberance. I then found that there are differences in the feathers of the wings in morphology, and those differences in males, and males produce non-vocal acoustic signals with their feathers when they're doing displays. And the differences in morphology account for the difference in the pitch at which the birds produce a sound. And then I did some computer simulations with the morphology of the migratory and non-migratory wings, and I found that migratory wings produce less drag, as you can see in these figures. So the angle of attack is basically what the angle at which the airflow is flowing against the model. And then the migratory birds produce less vortices, which is that gelatinous structure that you see around the wing. So likely the changes in behavior influence morphology because of natural selection, and that subsequently influences the communication signals. So we think that they are different species. Loss of migration promoted breeding allopatry and also changes in the breeding schedule, and then morphological evolution that leads to differences in communication signals. But this happened relatively a long time ago in microevolutionary terms. So we estimate divergence time of 1.4 billion years ago. So there are still some questions that I have regarding how the process happens at initial stages of divergence. And particularly for this talk, I want to focus on the role of gene flow on potential divergence of the species. So classically in birds, divergence is thought to happen geographically and where there's instantaneous divergence between the species. But I wonder if there's recurrent recruitment of migratory birds to the non-migratory population, and this process is more consistent with speciation with gene flow. So to approach that question, for my postdoc at Penn State with Dave Tefts, I started studying barn swallows. This picture is there, at least one. Okay, so this is the distribution of barn swallows in the American continent. So in aquamarine, you can see the breeding distribution of barn swallows. It's widespread across North America. And then it migrates to South America, and you can see it's also widespread in its overwintering range. What's cool is that in 1980, 6 nests were found under a bridge in Argentina, in the Buenos Aires province. And since then, there has been a very rapid expansion of breeding colonies. And this expansion has been well documented by Argentinian researchers. And you can see that it has expanded to the point where it's reaching the borders with Chile. It has also evolved a new overwintering area, which is shown that in that orange polygon in North and South America. So they don't go back to North America. And this has happened in 44 years. So the questions that I've been interested in working, some of the questions, is whether there is genetic differentiation that can happen in really fast time, and if there is a mutation driving this change of behavior, or if the genes are something that evolved later in this scenario. So what you have here is, what I'm showing here is a pairwise comparison between 3 different populations. So in the graphs you see in the x-axis is the position in the genome, the y-axis is a measure of genetic differentiation. And you can see that the 2 top panels are comparisons with Argentina, and the bottom panels are comparisons among North America, Eastern and Western North America. We did this with, we sampled blood samples in Argentina in 2 localities, and then we requested museum tissues for our populations in North America and used 1 published data set from Colorado. And what you can see is that the Argentinian populations are genetically differentiated from North American populations. And that's overall across the genome. We do not find indications of low genetic differentiation and 1 big peak of divergence, suggesting that genes were responsible for the behavioral change. So I think I got very excited about this because I wanted to understand how demographic processes affect genetic differentiation. And I think this is a really cool system in which we can ask these types of questions. So after a severe reduction in population size, the individuals that are born after tend to have long continuous homozygous regions in their genomes. Like, for example, in this study of gray wolves, each line represents a different individual, and then the panel on the bottom are individuals that are born from a small population, and the panel on the top are from mixed immigration coming from different populations. And in the lower individuals, you can see that the y-axis in this case is a measure of heterozygosity, and the x-axis is the position in the genome. And you can see that there are some individuals that have long continuous homozygous sequences, or runs of homozygosity. So there are some theoretical expectations of different attributes of runs of homozygosity under different demographic scenarios. So, for example, if there has been a bottleneck, you could expect that there would be many runs of homozygosity. So here in the lower graphs, n rho is number of runs of homozygosity. So you would expect many runs of homozygosity, but short because recombination breaks them down. If they are inbred, you would find a lot of runs of homozygosity, but very long. So this is what we find with our data. The North American population is shown in blue, and the Argentinian population is shown in red. And I guess this is not surprising that from a population that started with 6 nests, you would find inbreeding. But the North American population resembles an admixed population. The Argentinian population resembles one that has a lot of inbreeding. And when we look at the size classes of these runs of homozygosity, we find that some individuals in Argentina have very long runs of homozygosity, which are shown here on darker red colors. And then consistently, we find that heterozygosity in Argentina is lower than in North America. What's puzzling is that we don't find any differences in other measures of genetic diversity, particularly in allelic diversity. So pairwise allelic diversity and overall allelic diversity are not significantly different between Argentina and North America. And we started thinking whether gene flow can lead to this pattern. So North American birds have a really great resource, which is citizen science data collected through the North American Breeding Bird Survey. And we know that barn swallows have been declining, consistently declining since the 1980s. And so we can use this decline to inform demographic models. In Argentina, Cristian Torres, undergrad from the University of Rio Cuarto, has surveyed active nests since the first time that they were reported in Córdoba. And he surveyed them for 5 years. So we know how they have increased in that locality. And we use this rate of increase, which is exponentially really fast, to inform our demographic models that I'll show you next. So we run these models in fasting coal. They differ on whether they have gene flow or not. So the first model doesn't have gene flow, then bidirectional gene flow, and then unidirectional gene flow. And we find that the model with the highest support is one in which there is unidirectional gene flow from North America to Argentina. And the values of gene flow are low, so there are very few individuals that are bringing genetic diversity to the population. I do want to point out that I was very excited that these models estimated the time of divergence to be 51 years, which is very close to the actual date that we know was the first report of the nests. And this is a very nice concordance between empirical data and theoretical data and the power of coalescence theory. So to conclude this part of my talk, we find that switching hemisphere has promoted genetic divergence. They're very inbred, but it doesn't seem to be bothering them. They are ecologically very successful, and I define ecological success here because they are spreading their range, and they are also very abundant. And we think that gene flow may be bringing a little bit of that genetic diversity that they can use and reduce the likelihood of being extinct if something changes. I do wonder what's going to happen with the local species, where they're breeding in Argentina, and there's other species that breed there, and particularly the fork-tailed flycatcher, because they have very similar ecologies. In fact, we found that in eastern Colombia, where they winter, fork-tailed flycatchers and barn swallows roost together in ephemeral river islands of the Meta River, and they also forage together, so they're roosting and foraging. They are very social, both of them. They are incredible aerial flyers, and I wonder, if there's going to be some interactions that are going to be detrimental in the Argentinian breeding grounds. But to talk a little bit about these ephemeral river islands, because I'm very excited about this system. So if you go to the Colombian Llanos right now, it's going to be very flooded. It's going to be miserable with mosquitoes. Roads are difficult because there is a seasonality on rain. So the first panel up there is precipitation. And you can see that precipitation increases in the middle and then decreases. And on the x-axis, I labeled with blue when the river sort of like floods and there's no islands in the Meta River. And then in green is when the river comes down and the islands emerge. And then I included a panel of observations from eBird of yellow warblers. And you can see that the phenology of the birds coincides with the phenology of the river. And we found that this is the case for other austral migratory species, boreal migratory species. They are coexisting in these places and roosting in these ephemeral river islands. And so I wanted to point out that from this figure, 3 of these species have intraspecific variation in migratory behavior. And I want to briefly talk about the last point in my talk, which is moving a little bit outwards in scale and thinking about the fact that there are multiple species that have migratory behavior. So we can ask questions comparatively. And one simple question that I have is if there are patterns of distribution across species of migratory and non-migratory populations of the 900 species that have variation. So we can use bird life data to ask this question. For example, in this leaf warbler, the yellow distribution is the breeding distribution of migratory leaf warblers, and then the green distribution is the non-migratory breeding distribution of this species. Blue is where they pass through. And so we collected all the data for all the species, and we find some consistent patterns. So here you have a summary of all these, of the distribution of all these populations. In blue where there are more migratory populations. In yellow is where there are more non-migratory populations. And then the red or purple indicates areas where there's overlap. And there seems to be a line of overlap in the northern hemisphere. And indeed, we find that the point at which the species diversity or population diversity of non-migratory birds intersects with those of non-migratory coincides with the freezing line, which is the point at which environments turn from being subtropical to being temperate. Here we have latitude in the y-axis and then species richness in the x-axis. So we can start thinking about if we can predict under which conditions migration is turned on or off. And so I'm showing you here for a model for the whole 900 species using temperature seasonality as the predictive variable, and then whether migration is expressed or non-migratory behavior is expressed. But when we plot the curves for each individual species, we find that there's a lot of variation. So yes, there is a pattern of seasonality influencing whether they turn on and off or off, but there are likely other intrinsic traits of the species that determine whether they will migrate or not in a particular place in the world. And I think that this graph illustrates a little bit of how I think about, or how I approach this question. Give me 1 second, I have. So our barn swallow project, the backbone of our project is natural history observations. Without those, we couldn't be asking the questions that we are asking now. And I think that those natural history observations can feed this comparative work so that they are more informative. And with that, I want to say that I think switches in migratory behavior are an underappreciated mechanism that promotes species diversity on Earth. However, I should say there's a lot of research, very interesting research coming out. In this same session earlier, Nick Bone was giving a talk on variation in migratory behavior, and Gina Calabrese was giving a talk on whether migratory behavior influences speciation in different families of birds. And this is very exciting to me because it means that my field is moving and people are very interested in it, and there's a lot more that we're going to learn. This work is, of course, the work of many people. I want to acknowledge all my collaborators. They have been great. And I want to acknowledge my mentors because I've been very fortunate to work with great people. And I was very happy in my graduate studies and in my postdoc, and that's in part because of them. And I've been learning so much from them, and I hope I keep learning. And yeah, I really want to acknowledge that they are a big part of my work. I want to thank the American Society of Naturalists for including me in this symposium and everyone who made this talk possible. Also, museum collections. My research is based a lot on museum collections and also the funding agencies. I will be starting my lab in 2 weeks. So if any of you is interested or know someone who might be interested in this type of work, please come to talk to me or email me. And I think I have time for questions. Thank you.
I have one. And I can't remember which species because you talked about many cool species. The ones, large runs of homozygosity, but maintaining allelic richness. So doesn't that suggest what I've come to call population inbreeding, where they're mating with relatives more often than expected by chance rather than drift? Because that will cause homozygosity, and I think it'll cause the longer runs.
Can you repeat the last part of the question? I know you're asking about the runs of homozygosity.
Yeah. Is this not working?
Yeah, I don't think
Am I?
I'm hearing it. Yeah.
I gotta get that.
Oh, good. Yeah.
All right. Sorry. that I think the pattern is, I think, consistent with mating with relatives more often than expected by chance rather than small population drift. Because that mating with relatives won't change allele frequencies, so it won't deplete the allelic richness like drift will. But I think it also causes longer, I think you even said it causes longer runs of homozygosity. So is that, did they somehow change their behavior? Those are the six nests in Argentina, is that what they were?
Yes.
They were
yes.
just
well,
wanted
it's not
to-
from those birds. It's from birds from after the expansion. But I mean, it's
Back,
yeah.
okay. You think there was some change in behavior where they were more likely to mate with relatives than before? It seems consistent with that, but I could be wrong.
With that there is no bottleneck, no founder event, but big populations still recruiting. So I think that's an interesting possibility. That would be still consistent with gene flow coming to the Argentinian population. Sure. The estimated population size with our models is smaller in the Argentinian population than in the North American population. It's still larger than, a very small, more than, I think it's 3,000 or something like that, which is, big. So yeah, that is consistent. That would be, I guess, the question that I would have there is how is it that we find those large numbers of differentiation with FSD? Because it's very differentiated. If there was a lot of gene flow and that was consistent every year, individuals coming there, then we wouldn't find the differentiation that we find in FSD. But it is a possibility, yeah.
Cool, thanks.
Okay. Oh, hi.
Thank you, Valentina, for the great talk. So I agree with you. Like the system in Argentina is a great system. I was curious because in terms of phenotype, did you have time to actually look at if there was a shift in terms of phenotype for those specific individuals, and could you map some of the phenotype probably with a GWAS or something to try to understand some mechanism?
Yeah, that's a great question. Those are some of the things that we're planning to do. So this project was funded by my postdoc fellowship, so it's limited because for our GWAS we need a lot more. But we keep going every year to capture birds, and we're also starting to capture in the expansion front, particularly in Patagonia, so that we have a bigger picture of the population. And there's also studies that we're planning to do with museum samples from when the 6 nests were started nesting there. Something else that I didn't mention here is that another swallow, the clip swallow, is also doing this breeding in southern South America, but instead of having an expansion front, is having multiple little populations that don't seem to be doing very well, but they have been doing this since 2018. So we're doing also a comparative study between those two types of colonizations, one that's like, multiple different colonies versus one that is expanding from a reduction in, I guess, founder event, if that's the case. Thank you.
All right, yeah, thank you all for coming, and thank you so much to ASN for presenting me with this award. It's such an honor to have the opportunity to speak in this symposium alongside so many really talented researchers. So like many of you here today, I'm drawn to the fields of ecology and evolutionary biology because I'm fascinated by the incredible biodiversity that exists in the natural world. All across the planet, we see these amazing examples of adaptive radiations, often occurring on islands, where organisms have rapidly diversified from ancestral species to generate new forms that are capable of occupying novel habitat and exploiting new resources. My research uses genomic tools to better understand how biodiversity such as this originates and how it can be maintained for future generations to come, particularly in the face of environmental change. And of course, one of the main processes that generates this phenotypic variation that we observe is speciation, whereby one species or nested historical group, if you heard the keynote talk from yesterday morning, splits into two lineages that follow independent evolutionary trajectories. Speciation occurs when a population that's connected through genetic exchange becomes divided by a barrier to gene flow. And over time, these groups become increasingly genetically distinct until they form reproductively isolated taxa that can no longer hybridize to produce fertile offspring. In addition to speciation, another evolutionary process that generates this biodiversity we observe is local adaptation, whereby populations evolve traits that confer a fitness advantage in their local environment. So here you can see two populations of a plant species that are distributed along an elevational gradient. And due to differential selection from local environmental conditions on either side of this elevational cline, these populations have diverged from one another in traits such as flowering time. If gene flow between the populations is low enough, local adaptation can eventually lead to speciation. But throughout both of these two processes, speciation and adaptation, genetic variation is crucial for the evolution of genetic differences and reproductive barriers that keep these lineages distinct. And this genetic variation can come from a variety of different sources. So first, new mutations can provide the raw material needed for local adaptation and speciation to occur. However, for organisms with long generation times, it can take a really long time for relevant mutations that confer a fitness advantage to arise in wild populations. Alternatively, populations or species can adapt from standing genetic variation, or genetic variation that is already pre-existing within the group. And in contrast to adaptation from new mutation, adaptation from standing genetic variation tends to occur much more quickly, since it relies on alleles that are already available and present at higher frequencies within the population. And then finally, gene flow or introgression, the transfer of genetic material from one population or species to another through interbreeding, can also introduce genetic variation that can facilitate adaptation and speciation. So my research broadly focuses on the conditions under which adaptation and speciation occur, and the contributions of both gene flow and standing genetic variation to these two processes. However, my research interests have shifted a bit over the course of my career. So while I started off as an empirical researcher, primarily addressing basic questions in ecology and evolutionary biology, I've grown increasingly interested in applied conservation research and how we can leverage genomic data to guide conservation decision-making. So today I'll share two stories that kind of span the gamut of my research trajectory. I'll start off by sharing a study that I carried out as a PhD student, where I investigated the rapid evolution of pre-mating isolation in a group of birds known as southern capuchino seed-eaters. And then I'll shift gears to discuss some more recent work that I've carried out as a postdoc to investigate how migratory birds are responding to environmental change, and discuss the implications of our findings for the management of endangered species. All right, so first off, the rapid evolution of pre-mating isolation in the capuchino seed-eaters. The southern capuchino seed-eaters comprise one of the most rapid avian radiations on the planet, and they're somewhat of an enigma from an evolutionary perspective. Unlike many of the island radiations that I showed you earlier, capuchino seed-eaters have diversified throughout the grasslands of continental South America, where they lack any obvious geographic barriers to reproduction, and they largely co-occur during the breeding season, with up to 7 capuchino species breeding in Iberá National Park in northern Argentina. In addition, they exhibit remarkably low levels of ecological or genomic divergence. And then while the females of these various capuchino species are phenotypically indistinguishable, the males are divergent in both plumage coloration and song. Interestingly, iberaensis, or the Iberá seed-eater, which is a member of the southern capuchino radiation, was first discovered quite recently, only in 2001. And throughout its extremely restricted breeding range that you can see here in blue, it overlaps entirely with the boxantha, or the tawny-bellied seed-eater, which is the most abundant capuchino species that breeds in Iberá National Park. Very little work had been done to understand how the Iberá seed-eater came to form. So I spent multiple breeding seasons in Iberá National Park in Argentina, locating nests and collecting blood samples from adults and nestlings to track patterns of genetic exchange between Iberensis and Boxantha. And all of the genomic analyses that I carried out for this project were done in close collaboration with Leo Campagna at the Cornell Lab of Ornithology. Okay, so since evidences was only recently discovered, I was first interested in examining the degree of genomic differentiation between these two species and identifying the genomic regions that underlie their phenotypic differences. As I mentioned earlier, the males of these two species are divergent in both plumage coloration and song, while the females are phenotypically indistinguishable from an avian visual perspective. And when we compared whole genome sequence data from breeding adults of these two species, we found that mean genome-wide FST was only 0.006, which is remarkably close to 0. There were no fixed SNPs between these species, and the few SNPs that did differentiate the species were concentrated in 3 divergence peaks, which you can see here on scaffold 257, 430, and 762. Amazingly, these divergent genomic regions only contained 12 genes, 3 of which are known to be involved in the regulation of coloration. And so given that the few divergent genomic regions between these species appear to be involved in differences in male plumage, I was then interested in testing whether these genomic regions containing pigmentation genes are actually relevant for reproductive isolation. This is a challenging step that's often left out of many genomic studies. So to do this, I investigated the strength of assortative mating by running a principal component analysis on DDRAD data that I generated from all sampled adults and nestlings. And despite the extremely shallow degree of genomic differentiation between Evidensis and Epoxantha, you'll notice that males, females, and nestlings of these two species clustered together on this tree. So paired males and females, as well as their offspring, consistently were in the same side of this plot, providing no evidence of hybridization through either social or extra-pair mating. And this finding is especially surprising because these two species breed in local scale sympatry in Ibadat National Park, and they lack any obvious temporal or spatial barriers to reproduction. So in this map, you can see the spatial distribution of about 50 nests that I found over the course of the study. And as you'll notice, these two species hold neighboring territories during the breeding season, sometimes as close as 50 meters from one another, and they initiate nest construction at the exact same time. And so our findings suggest that differences in male plumage patterning and potentially song likely contribute to assortative mate choice and prevent interbreeding between the species. So to further test the importance of these two traits, male coloration and song, in pre-mating reproductive isolation, I carried out a behavioral experiment where I presented territorial males of Evidensis and Epoxantha with various combinations of conspecific and heterospecific song and plumage. So I had 5 different treatments, and I combined 3D printed models with song playback in order to test the male's behavioral responses. And during each trial, I recorded the number of flights and attacks at the mount and the amount of time that the male spent at various distances from the mount. In this video showing a typical male response to that conspecific song and plumage treatment, you can see that these mounts were wildly successful at eliciting aggressive responses. So this is a male Epoxantha responding to his own song and plumage. And obviously he's not very enthused about this potential intruder on his territory. So I generated a response intensity score by running a principal component analysis on these various behavioral response variables. And when I compared PC1 values across treatment groups, I found that males of Epoxantha as well as Evidensis exhibited the most aggressive responses to that conspecific song and plumage treatment, intermediate levels of aggression to the treatments with one conspecific and one heterospecific trait, and largely ignored the heterospecific cappuccino and control stimuli, which suggests that both male plumage patterning and song are used in territorial interactions and potentially female mate choice. So having investigated the genetic basis of phenotypic differences between these species and the contribution of phenotypic differentiation to reproductive isolation, we were finally interested in investigating the origin of this novel plumage phenotype of Evidensis in the context of the broader cappuccino seed irradiation. So to do this, we increased our whole genome sampling to include all 10 southern cappuccino species, and we generated phylogenetic trees for those regions encompassing the divergence peaks. So here you can see that Evidensis, which is shown in blue, shares that divergence peak on scaffold 257, which contained the pigmentation gene TOIRP1, with ruficollis shown in purple. However, or in contrast, melanogaster, shown in green, possessed a species-specific variant at that peak of differentiation. Likewise, Iberensis shares that divergence peak on scaffold 430, which contained the pigmentation genes HERC2 and OCA2, with 5 other cappuccino species. However, in this case, ruficollis possessed a species-specific variant at that peak of differentiation. And finally, when we compare Iberensis to all of the other southern cappuccino species combined, the same 3 peaks come up as differentiating Iberensis from the other species. And so we find that multiple cappuccino species share variants at these divergence peaks, but that Iberensis possesses these variants in a unique combination. This finding suggests that this novel plumage phenotype of Iberensis likely arose through hybridization between cappuccino species and the subsequent reshuffling of standing genetic variation, or genetic variation that already existed within this cappuccino radiation. This idea that the reassembly of old genetic variants into new combinations can facilitate rapid speciation and adaptive radiation was first proposed in a paper published in 2019. In this paper, the authors cite numerous other systems in which this speciation mechanism likely contributed to diversification, including Hawaiian silver swords, Darwin's finches, and Lake Victoria cichlids. However, with the exception of cichlids, cappuccino seed eaters remain one of the few systems with robust evidence to indicate that the reshuffling of standing genetic variation through hybridization can generate novel phenotypes that are then subject to sexual selection. So to summarize this first section of the talk on the role of gene flow and standing genetic variation in the evolution of pre-mating reproductive isolation in cappuccinos, we found that these 2 members of rapid radiation are only differentiated by 3 genomic regions that appear to underlie differences in male plumage patterning. Though Ipoxantha and Iberensis lack any obvious temporal or spatial barriers to reproduction, and the females are phenotypically indistinguishable, we documented complete assortative mating by these genomic regions. And we found that both male song and plumage patterning are likely involved in pre-mating reproductive isolation. And then finally, we found evidence that plumage differentiation likely originated through hybridization and the subsequent reshuffling of standing genetic variation, which generated novel phenotypes involved in mate choice. And so our results thus illustrate how new lineages can form and quickly become reproductively isolated from co-occurring taxa. All right, so I've shared some genomic insights with you into how biodiversity can originate and the contributions of both gene flow and standing genetic variation to this process. Now I'd like to switch gears and talk about how that biodiversity can be maintained in the face of environmental change. So in the second part of the talk, I'll focus on some more recent work that I've carried out to investigate adaptation to climate change in the southwestern willow flycatcher, which is an endangered migratory songbird, and the role of gene flow in that process. So as we're all aware, climate change is threatening the survival and reproduction of many species. And when faced with rising global temperatures, species can respond in 2 main ways. First, they can shift their distributions, moving to higher latitudes or up in elevation to escape warming conditions, or they can remain in place and adapt to changing environmental conditions through either phenotypic plasticity or genetic changes. This study focuses specifically on that second response, or adaptation through genetic changes. A species' ability to evolve phenotypic or molecular changes in response to altered environmental conditions depends on its adaptive potential, or the amount of genetic variation that's present in fitness-related traits. In a phenomenon known as the extinction vortex, population declines are often associated with higher levels of inbreeding and the heightened influence of genetic drift, which can lead to reductions in genetic diversity and the accumulation of deleterious alleles that negatively impact fitness and compromise the ability of populations to respond to environmental change. This, in turn, can further reduce population size in a positive feedback loop. However, one way in which small populations can escape from this extinction vortex is through genetic rescue, or the introduction of new genetic variation into a population that increases population fitness. Recent advances in our ability to sequence the DNA of historical specimens now allow us to track changes in adaptive potential over time and assess genomic responses to environmental change over the past century. So for this project, I used historical DNA sequencing to investigate adaptation to climate change in the willow flycatcher, which is a widespread migratory songbird. The willow flycatcher breeds across the United States and Canada, spends a non-breeding season from Mexico to northern South America, and is comprised of 4 different subspecies. Now, Extremis, or the southwestern willow flycatcher, has undergone really steep population declines and was listed as federally endangered in the US in 1995. So to investigate both past and present patterns of climate vulnerability in the southwestern willow flycatcher, we generated whole genome sequence data from 221 contemporary individuals that were sampled across the breeding range, and this included 18 birds that were sampled near San Diego in California. In addition, we sequenced 17 historical specimens that were collected between 1888 and 1909 during the breeding season near San Diego. The reason we focused on San Diego and Southern California for this study is because it's an area within the southwestern willow flycatcher breeding range that has undergone population declines, and also a location where we could obtain multiple contemporary and historical specimens in order to accurately estimate allele frequencies. And I should mention that the sampling design wouldn't have been possible without the contributions of many collaborators. So I'd just like to highlight Evan Paxton and Barbara Coos, both with the US Geological Survey, who contributed the majority of the contemporary samples for this project. Okay, so we generated genomic data from both the historical and contemporary samples at about 130,000 loci that were distributed throughout the genome. We ran an admixture analysis to examine whether population structure within San Diego has remained stable over the past century. So here, each vertical bar represents or depicts an individual, and the colors show the proportion of genomic ancestry from 4 different breeding clusters. As you'll notice, this admixture plot clearly pulls apart individuals that were sampled in the core range of the southwestern subspecies, here in orange, the eastern subspecies in blue, birds captured in the Pacific Northwest, here in green, and that historical population in gold. However, rather than forming its own discrete cluster, this contemporary San Diego population appears to be made up of a combination of genetic material, or genetic variation, from the desert Southwest, historical San Diego, and the Pacific Northwest, which suggests that population structure within San Diego has actually shifted over time due to introgression from neighboring populations. And this shift in population structure was accompanied by an increase in genetic diversity, an increase in effective population size, and a decrease in inbreeding within the San Diego population, all of which is consistent with the hypothesis that gene flow from neighboring populations in the Pacific Northwest and desert Southwest have introduced new genetic diversity into the San Diego population. And this finding was also supported by formal tests of introgression. Okay, so I've shown you evidence of past introgression into the San Diego population and an increase in genetic diversity over time. However, what does this past introgression actually mean for climate adaptation and adaptive potential in the southwestern willow flycatcher? To address this question, we downloaded high-resolution climate data from the historical past and the present, and we carried out a gradient forest analysis using all contemporary southwestern willow flycatcher samples, which provided us with a ranked list of climate variables that predict genomic variation within the southwestern subspecies. So taking the top ranking environmental variables, we find that TD mean, or mean dew point temperature, which describes the amount of moisture content in the air, and monthly precipitation during the breeding months are most strongly associated with genomic variation within that southwestern subspecies. Using this information, we then carried out 2 types of genotype environment association analyses that control for population structure. And we identified 232 candidate genomic loci that are linked to those top-ranking environmental variables. Okay, so earlier you might recall that I showed you this admixture plot of all loci, and I told you that population structure within the San Diego population has shifted over time to more closely resemble neighboring populations in the Southwest and Pacific Northwest. However, when we look at shifts in population structure at these climate-linked loci, we see a completely different pattern. So here you can see that, unlike the rest of the genome, climate-linked SNPs that were associated with mean dew point temperature as well as monthly precipitation actually shifted away from the desert Southwest and Pacific Northwest within the San Diego population over the past century. And this shift in population structure at climate-linked loci corresponded to an increase in adaptive potential over time. We also examined how allele frequencies of these climate-linked SNPs have shifted in response to changes in climate since the late 1800s. So taking a look at mean dew point temperature, the top-ranking climate variable from our gradient forest analysis, you can see that it has increased in Southern California over the past century. And if we examine how allele frequencies have shifted in response to this increase in mean dew point temperature, you'll notice that they tended to shift in the direction we would expect in order to adapt to changing climate conditions in Southern California. So in other words, alleles that were positively associated with TD mean tended to increase in frequency over time, while alleles that were negatively associated decreased in frequency within this population. Like mean dew point temperature, monthly precipitation I forgot to mention that the proportion of loci that shifted in a direction consistent with climate adaptation was significantly greater than expected by chance. Like mean dew point temperature, monthly precipitation has also slightly increased in the San Diego population. And once again, we find that allele frequencies have shifted in the direction we would expect in order to adapt that increase in monthly precipitation in the San Diego population at 51 out of the 72 candidate loci we identified. And so these findings indicate that this population is likely adapting to changing climate conditions in Southern California over the past century through some degree of genetic adaptation. So to summarize our findings on climate adaptation in the willow flycatcher, by comparing historical and contemporary samples, we found that the San Diego population of willow flycatchers was actually historically quite distinct from the rest of the endangered southwestern willow flycatcher breeding range. However, gene flow or introgression from neighboring populations into San Diego over the past century caused them to more closely resemble one another. This gene flow into San Diego was accompanied by an increase in effective population size and adaptive potential over time. And finally, we found evidence that this population may be adapting to the increase in mean dew point temperature and precipitation in Southern California. So while these alleles that were introduced into the San Diego population weren't necessarily adaptive at the time of introgression, the increased genetic diversity at both neutral and climate-linked loci through gene flow likely provided the raw material needed for this population to persist following population decline and adapt to changing environmental conditions. And so taken together, these results highlight how gene flow can increase evolutionary capacity and resilience to environmental change. And our results also support the idea that conservation intervention efforts aimed at maintaining genome-wide genetic diversity through strategies like genetic rescue and assisted gene flow could serve as important management tools to conserve vulnerable species in a changing world. In addition, our study has implications for how we perceive admixed or hybrid populations from a conservation perspective. Well, hybrids have long been undervalued in the field of conservation and are often perceived as posing a threat to distinct lineages. However, the results of both this and other recent studies highlight the potential conservation value of admixed populations, particularly when it comes to rapidly adapting to environmental change. Just to give one example, in a recent study by Chris Brower and colleagues, they found that hybridization between closely related species of rainbow fish that span an elevational gradient in Australia has likely improved their ability to cope with changing climate conditions. So while the upland species of rainbow fish have relatively restricted breeding ranges and lack the genetic diversity needed to persist in warm climates, hybrid populations between these upland and lowland species possess a greater frequency of warm-adapted alleles and have a reduced risk of extinction in the face of climate change. And so if we zoom back out to our original question about how biodiversity originates and persists in the face of environmental change, I think we've learned a number of key takeaways about the conditions that can lead to rapid speciation and adaptation. So first, while gene flow has traditionally been thought to disrupt adaptation and speciation, these studies indicate that gene flow and hybridization between distinct lineages can actually act as a creative force in the evolutionary process, introducing new genetic variation that can help organisms persist following population declines, and generating novel allelic combinations that natural selection can then act upon to drive the formation of new species and facilitate adaptation to changing environmental conditions. In addition, this work highlights the importance of maintaining the connectivity of populations that are adapted to different climate regimes and potentially carrying out intervention strategies that introduce new genetic diversity into vulnerable populations to aid with climate adaptation. And then finally, this research reveals the potential conservation value of hybrid or admixed populations. The advent of high-throughput genomic sequencing technology has revealed that hybridization is much more common in the evolutionary history of many species than we once thought. And by protecting admixed populations, we may be able to conserve some unique genetic diversity that may otherwise be lost due to environmental change. I have a lot of people I'd like to thank for this research, particularly my co-authors on both of these projects, including Scott Taylor and Leo Campagna on the Cappuccino work, Kristen Ruig on the Willow Flycatcher project, the museums that generously provided samples for our historical study, and of course the many funding sources and organizations that provided logistical support for this work. Thank you all so much for your attention, and I should have time for some questions.
Thank you. Thank you very much. So sort of a two-part question. Was San Diego historically particularly dry and warm? And I didn't catch, you may have said so, but were some of those candidate alleles in that historical population already? And was it more a function of the effective population size that you're positing as being important? Or was it actually new alleles coming from elsewhere?
We unfortunately, we don't have a great handle on that. I think likely there was genome-wide introgression that introduced like new alleles across the entire genome, and then selection probably acted on some kind of combination of standing genetic variation within the San Diego population, as well as introgressed alleles. We ran some formal tests of introgression, but unfortunately there were so few climate-linked loci that we couldn't really detect a strong signal in those introgressed regions. So yeah, the jury's out. We need more time points, honestly, for this population to figure out when introgression actually occurred and what it acted on. And then the other part of the question was about historical climate conditions in Southern California. So temperature is a little bit tricky because I think our data indicated that maximum temperature has actually decreased over time, maximum monthly temperature, but the mean temperature has increased. So there's kind of like conflicting signals there. And unfortunately, with the resolution of historical climate data that we can get from the late 1800s, it's restricted to monthly averages. But it would be interesting. I would be interested in looking at heterogeneity over time and whether fluctuations are increasing and how that may play a role.